Imagine a groundbreaking discovery that could redefine our understanding of human health and disease. Researchers at Penn State University have identified the first ever three-way relationship linking human genetic variation, the fungal component of the gut microbiome, known as the mycobiome, and the risk of chronic diseases. This breakthrough study, led by the Eberly College of Science, expands our knowledge of how human genetics and gut fungi interact, potentially paving the way for more personalized medical treatments. Gut fungi have been largely overlooked in comparison to bacteria and archaea, explains Emily Davenport, an assistant professor of biology at Penn State and one of the authors of the study. Our research reveals for the first time that human genetics significantly influences the composition of gut fungi, which could have profound implications for human health. This finding is crucial because it suggests that the diversity of fungi in our gut isn't just a matter of diet, but is also shaped by our genetic makeup. The study, published in the open-access journal PLOS Biology, involved a team of scientists from Penn State's One Health Microbiome Center, OHMC. They conducted a genome-wide association study, GWAS, to explore the links between human genetic variations and the presence of specific gut fungi. By analyzing data from 125 individuals, the researchers identified 148 genetic variants across 7 chromosomes that are associated with 9 different types of gut fungi. These genetic markers were then validated in larger datasets from the United Kingdom and a global consortium focused on coronary artery disease. One of the most intriguing findings was the connection between a specific yeast, Kazakhstania, and the risk of cardiovascular disease. This link is particularly significant and warrants further investigation, notes Seth Bordenstein, the Dorothy Fohuck and J. Lloyd Huck Endowed Chair in Microbiome Sciences and Director of OHMC. Understanding how human genetic variation affects the mycobiome could lead to new diagnostic tools and therapeutic approaches. But here's where it gets controversial. Some experts argue that the relationship between gut fungi and human health is still not fully understood, and more research is needed to confirm these findings. For instance, could the observed genetic links be influenced by environmental factors, such as diet or lifestyle, which were not fully accounted for in this study? Clinicians' ability to diagnose and treat chronic diseases is often hampered by a lack of clarity around the contributing factors, Bordenstein adds. By exploring the interplay between human genes and gut microorganisms, we hope to unlock new insights that could revolutionize personalized medicine. The research opens up numerous questions for future studies. For example, will similar genetic associations be found in different populations? How do gut fungi and bacteria interact? And how might these interactions be influenced by genetics? As Emily Van Sciyock, a postdoctoral researcher at Penn State and the first author of the paper, puts it, we are just scratching the surface of this complex relationship, and the possibilities for discovery are endless. The U.S. National Institutes of Health and Penn State provided funding for this research. With ongoing support, researchers at Penn State continue to tackle real-world problems that affect the health, safety, and quality of life of people globally. However, recent federal funding cuts pose a threat to this vital work. Learn more about the implications of these cuts at Research or Regress, https://www.psu.edu/research/real-world solutions. What do you think about the potential of this research to transform our understanding of human health? Do you believe that genetic factors play a significant role in shaping our gut microbiome? Share your thoughts in the comments below.